This is the lecture for Chapter 5 on membrane structure. One of the important parts of the cell theory, which I discussed in a previous lecture, is that all living things have cellular structure. And that includes, on Earth, the two types of cells, the prokaryotic cells and the eukaryotic cells. And these are diagrams of the two types of cells. And if you notice, both of them have a cell membrane, sometimes referred to as a cytoplasmic membrane or as the plasma membrane. And that is very, very important for all cells because cells need to be able to create compartments. So they need to be able to separate the inside, the cytoplasm of the cell, from the environment. And also in eukaryotic cells, you have the organelles, which are membrane-bound areas of cytoplasm that have a specific shape and function. And so that's very important for the cells to be able to create this barrier, to form these compartments, because it will allow for efficiency in metabolic processes and also their ability to respond to the environment quickly. Now, another thing about the cell membrane, especially in the eukaryotic cells and especially the animals, if there's any malfunction in the cell membrane, that can also lead to a disease process. So certain human diseases like cystic fibrosis, diabetes, cancer, these can all be traced back to defects or malfunctions of the plasma membrane or its components. This is a diagram of a typical animal cell membrane, and this shows you the basic structure of the membrane. And just for orientation, this is inside the cell and this is the exterior outside of the cell. As I go through this lecture, I'm going to go through the different components that you find in a typical animal cell membrane. And I'm going to start out talking about the phospholipids because they make up the main matrix of the membrane. I'm also going to mention cholesterol, which is an important component of animal cell membranes. Then I'll talk about what you find on the outside of the cell membrane. That would be the glycocalyx. And the last thing I'm going to go over are the proteins. And there are two types of proteins that you find in the cell. Peripheral proteins, which are found only on the inner layer of the cell, and then integral proteins, which span the whole width of the phospholipid bilayer. The phospholipid bilayer is the main matrix of the cell membrane. So the first thing to do is to review the basic structure of a phospholipid. So a phospholipid is a type of lipid that is composed of a glycerol, and the glycerol is a 3-carbon alcohol. So it's a 3-carbon alcohol, and for the phospholipid, one of the carbons has a phosphate group bound to it, so it has a phosphate group, while the other 2 carbons have fatty acids attached to them. And the fatty acids are nonpolar, so they are hydrophobic, and the phosphate head is hydrophilic. So the phosphate head is going to interact with the water, and the hydrophobic tails are not going to interact with the water. The other thing about the fatty acids is that they can be saturated or unsaturated. And the difference between the two is whether there is a double bond between 2 of the carbons. Unsaturated fatty acids have no double bonds, so you notice they're all single lines if you look at that structure. Whereas this other fatty acid has a double bond right there. A little bit hard to see, but it has a double bond there. And remember, double bonds are shorter, more rigid, so that produces this kink in the fatty acid. So this is one way to look at the structure. This is a space-filling model. So this is the polar head, and then the 2 nonpolar tails, and of course this one has a kink to it, and this is another way to denote it. So that's sort of the cartoon way that we often have in the book. So that's the structure of one phospholipid. When you take these phospholipids and you mix them into water, they spontaneously form a phospholipid bilayer. So, and this is due to the ability of water to organize nonpolar molecules, because remember, these fatty acid tails are nonpolar, they're hydrophobic. So what that means is when you mix them in water, the two layers, so the nonpolar tails on the two layers are forced together, and then the hydrophilic heads interact with water on each side. And then what ultimately happens is that you get a spherical structure. So you get a spherical structure formed in water. Water is on the inside, H2O, and water is on the outside. And then these hydrophilic heads are interacting with the water on the inside of this spherical structure, and then those nonpolar tails are in the middle, forced together in the middle. So anytime you mix phospholipids with water, because of the characteristics of the phospholipid, so it has polar head, nonpolar tails, and the ability of water to organize nonpolar molecules, you get these spherical structures, which is the basis for cellular structure. Within the phospholipid bilayer, so again, this is the phospholipid bilayer, the phospholipids do have the ability to move. But the most common type of movement is lateral movement. So lateral involves the phospholipids in one of the layers. So in one of the layers, these phospholipids can switch locations. So they can slide around each other, they can change places, and that happens only in one particular layer. So those phospholipids can move around each other and exchange places, and these phospholipids in the other layer can slide around and exchange places. And this happens a lot. So it's very fluid. You have a lot of movement of the phospholipids in their particular layer. Now, flip-flopping, going from one layer to another, can happen, but it's extremely rare. So this is not a common type of movement, whereas the lateral movement is very, very common. One of the things that can affect the fluidity of the membrane, that's how easily the phospholipids are moving, is whether the fatty acids are unsaturated or saturated. So unsaturated fatty acids have a lot of kinks, and so what that means is that those phospholipids cannot be packed in very tightly. So it ends up being more fluid. So the more unsaturated fatty acids you have in a cell membrane, the more fluid or the more movement you get in that membrane. So conversely, if you have a lot of saturated fatty acids, then they're very straight, they don't have those kinks, and the phospholipids can be packed in very tightly, and then you don't get a lot of movement. And we refer to it as viscous. So if a fluid is viscous, you mean it's very thick. It doesn't pour very easily. So the type of fatty acids that you have in the phospholipids, whether they're unsaturated or saturated, will affect the fluidity of the membrane. The fluid mosaic model is a term that is used to describe the fluidity of the cell membrane. Now, fluid, of course, is what I talked about before, how easily it is for the phospholipids to move in the plane of their layer. So again, not flip-flopping so much, but moving in their own plane. And mosaic relates to all the different molecules that you find in the cell membrane. This was a picture that I found that I thought demonstrated the mosaic aspect of the cell membrane really well. So this is a diagram of the cell membrane. Here is the phospholipid bilayer, and here are different proteins that are embedded in the phospholipid bilayer. So the idea, again, is that it looks like a mosaic, like a picture, like a mosaic, and that all of these molecules are fluid. They can move around in the plane of the membrane. Now, one idea that is modification of the fluid mosaic model is are called lipid rafts. Lipid rafts. And that idea is that not every individual molecule is moving on its own, but you actually have groups of molecules that are moving together. So instead of all these little individual phospholipids just swirling around on their own, you have groups, like possibly this red group is a raft and this blue group is a raft. So instead of moving independently, the whole group of molecules is moving around in the plane of the membrane. And then this blue group with that red protein in the middle is also moving around in the plane of the membrane. So those are called lipid rafts. And an analogy that I always think of when I think of the fluid mosaic model are these little games you might have had when you were a little kid or went to a fun fair and they passed these out. But it's plastic tiles that are all within one plane, and you're supposed to slide them around. So you slide them around, it mixes up the picture, and then you're supposed to slide them around to get the picture back again. But the point of this is you're not supposed to pop them in and out. They slide within a particular plane. And so that, again, is like those phospholipids. They can move fairly easily within their own plane. So these can move in their plane, and then these on the bottom side can move within their plane, but they don't pop in and out. One of the ways they identified that the fluid mosaic model is the process by which the membrane molecules move is they did an experiment where they took a human cell. So this was a human cell and it had all sorts of human proteins embedded in membrane and phospholipids associated with that cell. And then they took a mouse cell, which had mouse proteins embedded in the cell and phospholipids specific to the mouse, and then they fused them together. And what they ended up having was, I'll just use black here, a human mouse hybrid cell. And instead of just having all of the human proteins stay on one side and the mouse proteins stay on the other side, what they found is they all intermixed and eventually became evenly distributed. So that was the idea that sort of proved that there was a fluid mosaic model, that the proteins that are embedded in the phospholipid bilayer are not static, that they can slide around. And this is even important in, or even more important, in white blood cells. So you have your white blood cells that are meant to protect you from cancerous cells and also from cells that are infected. And what they noticed on the white blood cells is that white blood cells have certain receptors that are important for killing target cells. And when a white blood cell identifies a target cell, what it will do, so this is the white blood cell, is that it will take all of these receptors and it will move them so that they interact directly with the target cell, with the cancerous cell or the infected cell. And this is an example of a white blood cell. So this would be your white blood cell interacting with a cancer cell. So what it has done is it's taken all of those receptors that are involved in targeting the cancer cell, killing the cancer cell, and it's moved them all to the side to interact with the cancer cell. So the fluid mosaic model is very important in the function of cells because the cells can move the embedded proteins to the areas where they need to be. Cholesterol is an important component of animal cell membranes, and its purpose is to help maintain the shape of the cell and also maintain a constant fluidity for the phospholipid bilayer. So if you remember, animal cells do not have cell walls. So the purpose of the cell wall in a eukaryotic cell is to help maintain the shape of the cell. Since animal cells do not have a cell wall, then they need cholesterol to help maintain the shape of their cell. Also, cholesterol is going to maintain the fluidity of the phospholipid bilayer. So these are the cholesterol molecules, and they are a type of lipid, so made of rings. So they're aromatic. So they have a different structure than your typical triglyceride, which is similar to that phospholipid. So the cholesterol is embedded in the phospholipid bilayer, and again, this will maintain the fluidity of the cell of the phospholipid bilayer. So the thing about the cholesterol is that as temperatures increase, It will make the membrane less fluid, so it will maintain its structure. It won't fall apart. And then conversely, at low temperatures, if temperatures decrease, it's going to allow the membrane to be more fluid so it doesn't sort of freeze. It's going to maintain its fluidity so it can move the proteins to the area that the cell needs those proteins to be moved to. So cholesterol is a very, very important part of animal cell membranes to maintain the shape and the fluidity of the phospholipid bilayer. The glycocalyx is an important structure that is found on animal cells. And the meaning of the name is glyco refers to sugar and calyx refers to coat. So this is basically like a sugar coat that surrounds all animal cells. And sugar, of course, is a type of carbohydrate, so this is made of carbohydrates. Two basic types that you find are the glycoproteins, where the sugar is complex to a protein molecule in the cell membrane, or glycolipids, where the sugar is complex to a lipid in the membrane. And the glycocalyx surrounds the whole cell. So if this was your cell, you have basically this layer of carbohydrates all surrounding the cell. And the main purpose of the glycocalyx is adhesion. Because think of sugar, when it gets wet, it gets sticky. So the glycocalyx is what allows the cells to stick together. So it's going to help the cells stick together. All these carbohydrates are going to help them attach together. Also, the purpose of the glycocalyx is cell signaling. You can send signals between cells, or the cells can receive signals from other cells from far away. And it's also important for identification. So some of these molecules are used in cells being able to identify different cells. The extracellular matrix, or the EMC, is associated with the glycocalyx. And this extracellular matrix stands for extra, means outside of the cell, and it is a matrix of protein fibers that helps hold cells together. So that is the main function, to hold the cells together, but it's also involved in cell signaling and the movement of molecules into the cell. So this extracellular matrix is made up of protein fibers like collagen and elastin. So you've probably heard about those associated with skin, because this extracellular matrix is very important for animals because it holds the individual cells together into tissues so that the cells can coordinate their activity and function as a tissue. So you have collagen, elastin, you have proteoglycan, proteoprotein, glycan, carbohydrate, and fibronectin. Those are some of the fibers that you find in the extracellular matrix, and many of them are attached to integrins. Integrins are transmembrane proteins, integral proteins, and these proteins will bind to the integrins to hold the cells together. And they can also be bound to carbohydrates that you find in the glycocalyx. So again, the main purpose is attachment, holding the cells together so they can function as a tissue, but also they're involved in cell signaling, bringing molecules to send a signal to the cell, and also transport of molecules into and out of the cells. As I mentioned before, one of the important functions of the glycocalyx is attachment. So that glycocalyx, that sugar coat, can get sticky. Those carbohydrates, they can attach to each other and also attach to the extracellular matrix. And so all of that is important, especially in animal cells, to hold the cells together so they can function as a tissue. But another important function, especially in animal cells, is identification. So it's very, very important for animal cells to be able to identify each other. So to identify which individual they belong to, and also to identify that they're normal. So to identify if it's a normal cell or if it's a cell that has become cancerous or is infected with a virus, for instance. So there are specific molecules in the glycocalyx called HLA, and those molecules are used for identification. So HLA stands for human. The H is human, L is leukocyte, Leukocyte refers to white blood cells, and A is antigen. And an antigen is something that stimulates the white blood cells. So the HLA molecules, human leukocyte antigen molecules, are basically identifiers that you have on your cells. And so another way to look at this is it's like a fingerprint. Everybody, their cells, have their own unique combination of HLA molecules. So just as an example, there are about 11 genes that encode for the HLA, and they've been designated with letters A, B, C, E, F, G, and then the Ds, they have subsets of Ds. So you have DPA1, DPB1, and so on. So there are about 11 different genes that encode for these HLA molecules, and then the number of alleles. Alleles are different options for the gene. So like for eye color, there are two alleles, basically two alleles, brown eyes and blue eyes. So these are the different options for the genes. And there are lots of different options. So for A, there are about 4,000 options. B, there are 5,000 options. C, 3,000 options, and so on. So what this means is that there are a wide variety of combinations you can get with HLA. And so everybody has their own unique HLA composition. So just a little bit with Mendelian genetics. You have a father and a mother, and the father has his own unique set of HLA molecules. So he has two A's. He has A1 and A3, so those are the A part of his HLA, and he has two B's and then two DRs. So, and he has specific ones for that. The mother, she has her own HLA pattern. So she has A24 and A2, and then two different B's, two different DRs, and so on. So when they produce a child, if you remember Mendelian genetics, that child gets half its genes from its father and half its genes from its mother. So it is a combination. So the child's HLA is a combination of the fathers and mothers. So the thing where HLA comes into play a lot is if this child needs a transplant. So let's say that child needs a transplant, a kidney transplant. You need to do tissue typing to match the child's HLA to the donor. So you want to match it as closely as possible. So if you have a donor, you want the donor to have A1, A2, B8, B51, DR17, and DR11. The closer you match the HLA, the less likely you're going to have transplant rejection. Because remember, HLA stands for human leukocyte antigen. So these are molecules that could activate the white blood cells. So if the kidney does not match the child's HLA well enough, the white blood cells will be activated to reject that kidney. So especially in medicine, HLA is really important, especially in transplants and other processes and disease processes in humans. So far, in terms of the different components of the cell membrane, I've gone through the phospholipids, talked about the individual structure of the phospholipids with the polar heads and the two nonpolar tails, and discussed how when you mix them in water, they spontaneously form a bilayer with the nonpolar tails forced together and then the polar heads interacting with water either outside of the cell or inside of the cell, the cytosol. I've also gone over the cholesterol. Cholesterol is a type of lipid that is embedded in the phospholipid bilayer. In animal cells, it helps maintain the shape of the cell and also the fluidity of the membrane. I also went through the glycocalyx sugar coat. So this is a layer of carbohydrates that you find surrounding on the exterior of the animal cell, and it's important in attachment, especially to the extracellular matrix, and also for identification. All right, the last thing I want to talk about are the proteins. And there are two types of proteins, peripheral proteins. Peripheral proteins are found on the inside of the cell membrane, on the inside of the cell. And they can be used for attachment, attaching to the cytoskeletal fibers, or they can be enzymes, catalyzing reactions. So those are the peripheral proteins. But the main protein I want to talk about now are the integral proteins. And integral proteins are found inside, embedded in the cell membrane. Sometimes they're called transmembrane proteins because they go through the whole membrane, and they will have two hydrophilic parts, hydrophilic interacting with water. So they'll have one end that is hydrophilic interacting with water outside of the cell, and the other end is hydrophilic interacting with the water inside the cell. But they also have a hydrophobic region, and the hydrophobic region is interacting with those non-polar tails from the phospholipids. So that allows these transmembrane proteins, integral proteins, to remain embedded in the membrane because they have those different regions. And integral proteins can have lots of different functions, which I'll go through in a minute. They can also be attached to the extracellular matrix, or they can be attached to cytoskeletal fibers to hold them in place. First type of integral protein or transmembrane protein that I want to talk about are the channel proteins. And channel proteins are involved in transport. So transporting molecules that are too big or too charged to cross directly through the phospholipid bilayer. So these molecules cannot cross through the phospholipid bilayer, so they need to use a channel protein. Sometimes we call them in general transport proteins. And so a channel protein basically kind of looks like a tunnel. It's open and it will allow these molecules to transport in. And these molecules move in relation to the concentration gradient. So that's related to diffusion, which I will talk about. So these molecules are going to move from an area of high concentration to an area of low concentration. And as long as the channel is open, as long as this is open, they can move freely without using energy into the cell. Another type of transport protein are carrier proteins, and they're a little bit different than the channel proteins because they have to change shape to transport the molecules across the membrane. So again, this is involving molecules that are too big or have too much of a charge to cross directly through the phospholipid bilayer, so they need a protein carrier to carry them across. So instead of being like a tunnel, like the channel proteins, this is a carrier protein which sort of has to physically interact with the molecule, change shape to allow it to cross over the membrane. And again, this is due to a concentration gradient. So these molecules will move from the area of high concentration to the area of low concentration if the carrier protein is able to physically change shape and transport them across the membrane. Another type of transmembrane protein or integral protein that you will see in cell membranes are cell recognition proteins, and these are like those HLA molecules that I was talking about. So these are transmembrane proteins with carbohydrates attached to them. And again, there are many different types of these HLA, human leukocyte antigen molecules, that you find on cells. And just as a reminder, that those cell recognition proteins, the ones you have on your cells, it's a combination of ones you got from your mother and ones you got from your father, and you have a unique pattern of them. So again, it's kind of like a fingerprint that your body can use to recognize normal cells that should be in your body. A group of really important transmembrane proteins are the receptor proteins, and the receptor proteins are proteins that receive signals from other cells and then transmit that signal into the cell. So often these signals are molecules, and the most common ones are hormones, all sorts of different types of hormones. So in the human body, hormones are used as communication between cells. And so what basically happens is that the hormone will bind to a receptor that has a specific shape that binds only that particular hormone. And once they bind together, then that sends a signal into the cell so the cell can have a response. Signal transduction is the process by which a cell receives a signal and then transmits that signal to the inside of the cell to generate a response. So this process involves first, with a signal molecule, binding to a particular receptor, and then that transmembrane protein catalyzing a reaction, and often it involves several steps, and this is called the transduction pathway. So different proteins are activated, and that leads to some sort of response, and that could be targeting a structural protein to alter the shape or movement of the cell, could be activating an enzyme that will change metabolism in the cell, or activating a gene, a regulatory protein that will allow for the expression of a new gene. So all of this is a very important function to the cell. And a lot of human diseases occur when there is a mistake in this process, where there's either an inability to recognize the signal molecule, so you can't generate a response, or a mistake somewhere in the signal transduction pathway, or a mistake in the targeted protein. So almost every human disease can be traced back to a disorder or a malfunction of the signal transduction pathway. Another type of transmembrane protein are enzymatic proteins. And so these are proteins, transmembrane proteins, but their main job is to catalyze a reaction, convert a substrate into a product. And we'll talk about enzymes a lot later in the semester. The last group of transmembrane proteins that I want to mention are junction proteins. And junction proteins are used for attachment and communication. So these are proteins that interact with other proteins in other cells to either allow them to bind together or allow communication between cells. There are three types of junctions that you find in animal cells, and those are the adhesion junctions, tight junctions, and gap junctions. And they have slightly different functions in the animal cells. Adhesion junctions are involved in mechanical strength, so holding cells together. And these are often called desmosomes. And these are found in tissues that involve a lot of mechanical stress. So like the heart or the other muscle cells that are moving a lot, you don't want those cells to fall apart. You don't want the tissues to fall apart, so you have adhesion junctions. And they're also found in the bladder, which will expand to hold the urine, and so that's a lot of mechanical stress. So you have adhesion junctions. Tight junctions produce a waterproof tissue, so you're going to prevent fluid leakage. So this is what you find in the stomach, between the cells in the stomach and the bladder. So that's where you have structures that are holding a fluid that you don't want to leak into the rest of the body. And the last type of junction are the gap junctions. Gap junctions are used for communication. So this is between cells, and it allows molecules to pass from one cell to another to facilitate communication and coordination of tissue activity. In plants, the type of junction you find are the plasmodesmata, and the plasmodesmata are used for communication. So it's basically areas between the cell walls that allow molecules to pass back and forth to allow that communication and coordination between plant cells. In a previous lecture, I discussed why cells are small, and that has to do with the surface area to volume ratio. That is much better to have a high surface area to volume ratio. That means more surface area compared to the volume. And the reason for that is that the surface area represents how that cell interacts with the environment. So its ability to take nutrients in and to release waste products. So that's the surface area. Now the volume, the volume represents all the metabolism that is going on inside of the cell. So all the metabolic reactions, all the biochemistry. So for a cell to function, it needs to have sufficient surface area to allow for enough nutrients to come in and to release all the waste products in order to support the biochemistry, the metabolism that's going on in the cell. So for a cell to be good and functional, you want a very high surface area to volume ratio. And if you have a cell this large, one individual cell that large, you only have 5,400 micrometer squared of surface area. If you break that structure down into smaller cells, you end up having a lot more surface area, almost 3 times as much. So the smaller the cell is, the more transport you get out of the cell and into the cell, and the more transport you have, the better able you are to support the metabolism of the cell. So that's why cells are small. So that's just a review of why cells are small. The cell membrane, major function of the cell membrane, is to control that transport. So it's the membrane that is going to control whether molecules go into the cell or whether they are able to leave. And that can involve the phospholipid bilayer itself, or more often than not, different types of transport proteins. Cell membranes are considered selectively permeable barriers. So they are a barrier because they are separating the inside of the cell from the environment, but they are selectively permeable because they will allow some molecules to cross either into the cell or out of the cell. Now, there are many different types of molecules that may need to be transported into the cell or out of the cell, and depending on the characteristics of the molecules, that depends how they get in. There are a few types of molecules that have the ability to cross directly through the phospholipid bilayer, but there aren't very many of these molecules. These molecules have to be very, very small or non-charged, so non-polar. And the reason for this is because, remember, these hydrophobic tails are creating a large non-polar region. So in order for a molecule to be able to cross it, it has to be non-polar or very, very small. And some examples of things that can cross directly across the phospholipid bilayer are gases like carbon dioxide and oxygen, and also alcohol. So when those things cross, they move according to the concentration gradient. So they go from an area of high concentration to the area of low concentration across the membrane. Now, most molecules are not able to do this. Most molecules either have a charge or they are very large, like the macromolecules. So because of this, they cannot directly cross the membrane, so they need some way to be transported across. One example are channels. So I talked about the channels before, kind of like tunnels that allow molecules to cross directly through the membrane. And one such channel are the aquaporins, and aquaporins allow water. So aqua, water, they allow water to move across in either direction depending on the concentration gradient. And this will become very important when I talk about osmosis in a little bit. So just remember, though, that water molecules do not cross directly through the phospholipid bilayer. They need these channel proteins to cross, called aquaporins. And so other molecules that are large, like glucose, will need, or ions, they will need either channel proteins or carrier proteins. Then sometimes there are very large molecules, like the macromolecules, which in the molecular world are very, very large. They will need other forms of transport, such as endocytosis and exocytosis. And I'll talk about all those different types of transport next. The first type of transport that can occur across the cell membrane is called passive transport. And passive transport works along the principles of diffusion. So you should know already the basics with diffusion. Diffusion is a type of transport that requires no energy. No ATP is needed by the cell for this to happen. Also, it moves down a concentration gradient, so it moves from an area of high concentration to an area of low concentration. And the last thing is it can reach equilibrium. And those are the principles of diffusion. So the example of diffusion is if you have a beaker of water and you drop a drop of dye into that beaker of water. So you're going to have an area of high concentration of the dye, and that will move out from the area of high concentration to the area of low concentration. And you will get this movement until you reach equilibrium. Equilibrium is where you no longer have a concentration gradient. So the concentration is equal throughout the whole beaker. Now, the reason that you get movement of molecules in diffusion is due to Brownian motion. And Brownian motion explains the inherent motion of atoms. So in this diagram, I have the 4 atoms that make up living things: hydrogen, oxygen, nitrogen, and carbon. And you should know their atomic numbers. Hydrogen has the atomic number of 1. Oxygen has atomic number 8, nitrogen is 7, and carbon is 6. And so you should know their basic atomic structure with the protons and neutrons in the nucleus and then the electrons orbiting around the nucleus. All right, so that's the basic atomic structure. So in an atom, what is moving? It's the electrons that are moving. They are spinning around the atom. And so because of this, because you have this motion of electrons spinning around the nucleus of the atom, that gives the atom a little bit of an inherent motion. So sort of like a little jiggle that of course we can't see. But the analogy I think of is a top. If a top is spinning very, very fast, it seems to stay in one spot, but if you look at it closely, there's a little bit of movement around it. It sort of jiggles a little bit. It doesn't stay stationary. And that's the emotion, the Brownian motion that atoms have. So because of this movement of the electrons around the nucleus, every atom has a little bit of inherent motion. And so molecules, because they are made of atoms, they also have inherent motion. So if you were able to look at a molecule in a gas or a liquid, you would see that it kind of randomly moves around. So it randomly moves around kind of like a top would if you had a top out on a table. So it's randomly moving around, and that's just the inherent motion of molecules in a gas or a liquid. And again, it is all due to just those electrons spinning around the atoms. So this is random movement. It doesn't have a purpose. It's just that sort of little jiggle that these atoms, these molecules will have. And that is the movement that drives diffusion. Facilitated transport is a type of passive transport. So it follows the same principles as diffusion. So no ATP is required. You'll have the molecules moving from an area of high concentration to an area of low concentration until you reach equilibrium, where you have no longer a concentration gradient. There's no area of high concentration or low concentration, but the molecules still move due to that Brownian motion, but you don't have a net gain or loss. And facilitated transport is called facilitated because these molecules that are going to be involved moving across the membrane are too large to simply cross the membrane. So they can't simply diffuse across the membrane like carbon dioxide or oxygen or alcohol could. So because they are too large or they have a charge, they need some sort of transport protein. And this can involve a channel. So a channel, remember, kind of looks like a tunnel, allowing the molecules to move across. Or it can involve a carrier protein. Carrier protein has to change shape to sort of chaperone the molecules across. But either way, the direction of movement is going to go from an area of high concentration to an area of low concentration until you reach equilibrium. Now, facilitated transport can be stopped because if the carrier protein inactivates or changes shape, then it will no longer be able to transport the molecules across, and you won't have transport. Same with the channel protein. So channel proteins could be closed off, and then you don't get transport. That's different than diffusion. Diffusion, the cell cannot control it. So the cell can't control the movement of molecules with simple diffusion, but it can control facilitated transport. Osmosis is a very particular type of transport regarding a membrane, and osmosis can be defined as the diffusion of water across a semi-permeable membrane. So these three components are very important. For osmosis to occur, that means you must have a membrane, a semi-permeable membrane, and you are only observing the water molecules. So you're just observing the movement of the water molecules, not salt, not sugar, not gases, just water. And those water molecules are going to move according to the principles of diffusion. So no energy is involved. Movement is going to be from an area of high concentration of water to a low concentration of water until equilibrium is reached. Once you reach equilibrium, then osmosis will stop. Now, the important thing about osmosis is that you are dealing with solutions. So a solution is a combination of a solute and a solvent. So the solvent is the liquid. So this is the main liquid matrix, and the solute is what is dissolved into the liquid. So in biology, the solvent is always water. So it's always water in biology. If you go into chemistry, it can be other substances like alcohol. But in biology, it's always going to be water. So water is the solvent, and then the solute is anything you have dissolved in the water. So it could be sugars, it could be salts, it could be proteins, anything, gases, anything. So together, that equals the solution. Now, solutions are always expressed in percentages. So the solution itself will be 100%. So that means the percentage of the water plus the percentage of the solute will equal 100%. So if you have a 5% sodium chloride solution, then the water needs to be 95%. So solutions are always described in a percentage, and usually they're using the percentage of the solute. So a 5% sodium chloride solution, a 16% glucose solution, that sort of thing. When dealing with osmosis, there are three important terms that are used, and those terms are isotonic, hypotonic, and hypertonic. Now, these terms are used to compare two solutions to each other. So remember, osmosis is defined as the diffusion of water across a semi-permeable membrane. So that membrane is dividing or separating two solutions from each other. So these terms, isotonic, hypotonic, hypertonic, are comparing those solutions to each other. And the basis of comparison is the solute concentration. And that's what tonic refers to, the percentage of solute you have in the solutions. So isotonic, hypotonic, hypertonic are comparative terms comparing solutions in terms of their solute concentration. So an analogy could be height. So if you have a person and this person has a particular height. Now you can compare him to other people. And when you compare him to other people in terms of height, he could have either the same height, he could be shorter, or he could be taller. And the comparison, the word you would use depends on who you compare him to. So if I take this guy and I compare him to this woman, he is taller. So that's the comparison term, comparing two people to each other. He is taller, she is shorter. So that's the comparison term. But if I compare him to somebody else, if I compare him to somebody who is taller, now he becomes shorter. So it's very important to know what you are comparing. So if, for example, I gave you a beaker of solution, and if I told you it's hypotonic, your first question should be compared to what? Just like if I gave you this person and I said he's shorter, your first question should be compared to whom? Because he is shorter when you compare him to certain people, but he's taller when you compare him to others. So you need to really keep in mind that these terms, isotonic, hypotonic, hypertonic, are comparative terms. Now, the normal situation in biology when we're dealing with osmosis problems, The two solutions we are comparing is the environmental solution, so the environment, so that's solution A, and often we are comparing it to the cytosol in a cell. So a cell that is in the environment, and this is solution B. That's supposed to be a B. B. And of course, the cell membrane is the membrane separating the two solutions. So in biology, that's the usual situation that we are involved in. We're comparing the solution in the environment to the solution inside the cell. So that's the comparison. Now I want to go through these three terms step by step. And the first important thing to remember is that you are comparing two solutions to each other. So two solutions. And in this diagram, you have a solution on side A and you have a solution on side B, and they are separated by a semi-permeable membrane. So solution A and solution B. So isotonic. Iso means the same, tonic, solute concentration. So same solute. Solute concentration, and usually, again, we write it in percentages. So if a solution is isotonic to another solution, that means they have the same solute concentration. So that could mean that solution A on side A is a 5% sodium chloride solution, and it could mean that solution B is a 5% glucose solution. So it doesn't have to be the same solute. What's important is the percentage. So if they have the same solute concentration, that means they have the same water concentration. So that would mean that solution A is 95% water, solution B is 95% water. And now, because they have the same water concentration, there is no concentration gradient. Concentration gradient. There's no difference in concentration, so they are at equilibrium. Equilibrium. So there is no osmosis. No net movement of water. So because they are at the same concentration of water, there is no net area of high water or low water, and so they are at equilibrium. Now, water molecules will still cross the membrane, but they will cross the other way. There will be no net gain or loss of water. So that's when the solutions are equal to each other. They're considered isotonic to each other. So if the two solutions do not have the same concentration of solute, so if they are not isotonic, then that means one has to be hypotonic and the other has to be hypertonic. So in other words, or with the analogy of the height, if two people are not the same height, then one person has to be shorter and one has to be taller. So in this case, I want to describe the hypotonic solution. So remember, we're still comparing two solutions to each other, solution A and solution B, and now they are not equal in solute concentration. So they are not equal. So let's say side A is 5% solute and side B is 10% solute. So they're not equal to each other. So the hypotonic solution, hypo means lower, and tonic means solute. So the hypotonic solution is going to be the one with the lower solute. So that'll be the one with the lower concentration of solute. And the one with the lower concentration of solute is by definition going to have the higher concentration of water. And you can do the math on this. So if this is 5% solute, doesn't matter if it's sugar or salt or whatever it is, then it will be 95% water. And then this solution, if it's 10% solute, sugar, salt, whatever it is, then it's going to be 90% water. So the solution that has the lower concentration of solute is going to have the higher concentration of water, and it will always be that way. So that's the hypotonic solution. Again, if the solutions are not isotonic to each other, then one has to be hypotonic and the other has to be hypertonic. So as I mentioned before, in the example before, solution A was 5% solute and solution B was 10% solute. And so if they're not equal, which these are not equal, then one has to be hypotonic, one has to be hypertonic. So hypotonic is the one with the lower concentration of solute. So this was the hypotonic solution. So hypertonic solution, obviously the other has to be hypertonic, but going through the definition, hyper means higher. Higher concentration of solute. So, and just to double check, you have 5% solute here, 10% solute here. So the solution with the higher solute has to be the hypertonic solution. Hypertonic. And just as further comparison, if it has the higher concentration of solute, it has the lower concentration of water, solvent. Lower concentration of water. And if you go through again and calculate the water, if this is 5% solute, it's 95% water. If this is 10% solute, it is 90% water. So again, hypertonic solution, higher solute, 10 is higher than 5, lower water, 90 is lower than 95. So that's how you designate which solution is which. If they're not isotonic, if they're not equal in solute concentration and water concentration, then one has to be hypotonic. The hypotonic solution has the lower solute, higher water, and the other has to be hypertonic. Hyper, higher solute, lower water. All right, now when you have one solution that's hypotonic and one that is hypertonic, now you have a concentration gradient. Concentration gradient. So in terms of water, if we're looking at osmosis, you're concerned about the water. So if you're looking at osmosis, now you have an area where there's 95% water and 90% water. So now that you have the concentration gradient, now you're going to have osmosis. And osmosis, remember, is the diffusion of water across the semi-permeable membrane. So here is your semi-permeable membrane, selectively permeable membrane. You have a concentration gradient. Diffusion moves from the area of high concentration to the area of low concentration. So the direction of osmosis is going to be from the hypotonic solution to the hypertonic solution. And it always moves that way because the hypotonic solution is always going to have the higher concentration of water and the hypertonic is going to have the lower concentration of water. So if you determine which solution has the higher concentration of water, then you have figured out the direction of osmosis. And if you can determine which solution is hypotonic or hypertonic, then you have figured out the direction of osmosis. So osmosis always goes from hypotonic to hypertonic. There are several different ways to present problems involving osmosis, and so I'm going to give you 3 problems here in this video. So what you should do is read the problem, pause the video, see if you can solve it on your own, and then play the video again and see how I solve it. So the first question, a cell that is filled with 5% sodium chloride solution is put into distilled water. What will happen to the cell? All right, so this is how you could go about solving this. So what information do you have? You have a cell is filled with 5% sodium chloride solution. So this is your cell, the white cell in the blue beaker. So this has 5% sodium chloride, which means it must be 95% water. So 95% water, so that's what you know. And then you know the environment, it is put into distilled water. And distilled water is basically thought of as 100% water. So first thing you notice is they do not have equal solute concentrations. So if this is 100% water, it's 0% solute. So 0 and 5 are not equal. So these are not isotonic to each other. So since they are not isotonic to each other, you have to figure out which solution is hypotonic, which is hypertonic. So hypotonic. Remember, hypotonic, hypo means lower, like hypothermia, lower temperature. Tonic refers to solute. So which one has the lower solute? Well, 0 is lower than 5. So the environment is hypotonic. And then just to double check, you need to figure out which is the hypertonic solution. And remember, hypertonic, hyper, like hyperactive, a person who is hyperactive has a higher level of activity. So hyper means higher, tonic means solute. So which one has the higher solute? 5 is higher than 0. So the cell has the hypertonic, is the hypertonic environment. All right, now conversely, remember, hypotonic, lower solute means higher water. So higher water, which is higher, 100% or 95, 100% is higher, and that's still the environment, so the solution in the beaker. Hypertonic has higher solute, which means lower water. And which solution had the lower water? 95 is lower than 100, so it's still the cell. All right, so you have a concentration gradient, so you will have osmosis. What is the direction of osmosis? It works on the principle of diffusion. So the water is going to move from the area of higher water concentration of water to the area of lower concentration of water. So from the environment to the cell. So the water, the direction of osmosis is into the cell, and the cell will swell, and if it swells enough, it could lyse. So what will happen to the cell? Osmosis, the direction of osmosis is into the cell. Water will move into the cell, and the cell could lyse. Second problem. A cell that is filled with a 5% sodium chloride solution is put into a 10% sugar solution. What will happen to the cell? So again, what you should do is figure out the relative concentrations of the two solutions. Remember, one solution is the environment, the other solution is the cell. So you go through them, figure out the relative concentrations of the solutes and the water, determine if they are isotonic, or if they're not isotonic, then they're hypertonic or hypotonic, and then figure out the direction of osmosis. So A is the 10% sugar solution, 10% sugar. If it's 10% sugar, that means it's 90% water. B is the cell. B is the cell. It's 5% sodium chloride, so it's 5% salt. And again, the type of solute doesn't matter. What's important is the percentage. So it's 5% salt, so it's 95% water. So isotonic means they have the same concentration of solute. 10 is not the same as 5, so one has to be hypotonic, one has to be hypertonic. Hypotonic means lower solute. 5 is lower than 10, so this is hypotonic. And a hypotonic, if it's lower solute, that means it is higher water. (95) is higher than 90, so this is the hypotonic solution, and then by process of elimination, this is the hypertonic solution. Hypertonic hypertonic solution. And then just to double check, hypertonic, higher concentration of solute, higher concentration of solute. Relatively speaking, it'll have the lower concentration of water, and 90 is lower than 95. So this is your hypertonic solution. This is your hypotonic solution. You have a concentration gradient. Osmosis always goes from hypotonic, higher water, to lower tonic. So hypotonic to hypertonic. So water is going to move from B to A. Water is going to move from B to A. The direction of osmosis is out of the cell. The cell is going to lose water, and if it is an animal cell, we call it plasmolyzing. So it's going to dehydrate, it's going to shrink, and if it loses enough water, it's going to die. The last practice problem for osmosis is when a cell is placed in a sugar solution, it gains water. Explain what happened. So for this problem, I'm not giving you percentages, but I'm giving you the effect, what happened. And what happened is that the cell gained water. So the first thing you know is that a cell is placed in a sugar solution. That means there is some sort of percent of solute in the environment. Okay, so you have a percent of solute in the environment. And now you know that the cell gained water, so you know the direction of osmosis was into the cell. So that tells you that the solutions were not isotonic. If they were isotonic, they would have had the same concentration of solute, same concentration of water. There would have been no net gain of water, and so no osmosis would have occurred. So you know there was osmosis. You know they were not equal. So that means one solution was hypotonic and the other solution was hypertonic. And you know that hypotonic solutions have the lower solute, which means they have the higher water concentration. And the hypertonic solution, conversely, has higher solute and lower water. So the direction of osmosis is always from the area of higher water concentration to the area of lower water concentration. So it always goes from the hypotonic solution to the hypertonic solution. So if the direction of osmosis was into the cell, then that means the hypotonic solution was the environment. So the hypotonic solution was the environment and the hypertonic solution was the cell. So you know that the cell was hypertonic compared to the environment. So you know that the cell had a higher concentration of solute than the environment and a lower concentration of water than the environment. So far, I have discussed passive transport, and passive transport has involved simple diffusion, facilitated transport, and osmosis. All of those things did not involve energy. It involved molecules moving down a concentration gradient and would end when it reached equilibrium. Second general type of transport you find in cells is active transport. And active transport is very different because active transport does require energy. It requires the cell to use ATP, the readily usable form of energy in a cell. The other difference with active transport is now you are moving up a concentration gradient. So you're moving molecules from a low concentration to a area of high concentration. And so that you are basically building up a high concentration on one side of the cell. And because of this, because you're going from low to high concentration, you will never reach equilibrium. You're actually going in the opposite direction. You're making more of a concentration gradient. This is an example of active transport. So this is an example of the sodium-potassium pump. And this is called a pump. Often they're called pumps because they require ATP. So the cell requires ATP to facilitate this transport. And it also involves some sort of carrier molecule. So transport molecule, specifically a carrier molecule. And in this case, the sodium and potassium is linked. So for this example, first sodium is going to bind to the carrier molecule. So sodium is going to bind to the carrier molecule, cause it to change shape, and release the sodium outside. So it's building a high concentration of sodium on the other side of the membrane. And once it transports the sodium, then it's going to transport the potassium. So the potassium is going to bind, change the shape of the carrier protein, and release the potassium inside the cell. So this is kind of a complicated transport. So again, it is involving ATP, so it's an active transport, and it's moving molecules from an area of low concentration to high concentration. So you're not going to reach equilibrium. Now, sodium-potassium pumps are really important to the function of certain cells in the human body. For neurons, it's very important for the action potential. So the action potential in the neurons involves sodium and potassium changing the concentration, and that is transporting the nerve impulses. So from your body to your brain, and then the nerve impulses back down to your body for a response. Also, sodium and potassium is involved in muscle function, so muscle contraction. So it's very important to have a difference in concentration of the sodium and potassium to facilitate muscle contraction. So this is a very, very important type of transport, especially for the human body. The last type of transport that can occur is bulk transport. And when we talk about bulk transport, we mean transport of very large things. It could be macromolecules like carbohydrates or proteins, or it could be things as large as other cells. So bulk transport is involving lots of very large molecules that cannot cross the membrane even using a transport protein. So when you have bulk transport occurring, it expends a lot of energy because the cell needs to move its membrane to form vesicles. Now there are two types of bulk transport. First type is exocytosis. Exocytosis. And exocytosis means that the molecules are being moved outside of the cell, exo. So this is the inside of the cell. This is the plasma membrane. Inside the cell, there's a vesicle that contains some sort of molecules that need to be released outside of the cell. So for instance, these could be hormones, protein hormones that were made in the rough ER. They were modified into a functional form in the Golgi apparatus, and then they were packaged into a vesicle. So a vesicle is basically a little membrane-bound area. So here are the protein hormones, and they need to be released outside of the cell into the blood so they can go interact with the receptors of other cells. So to do this, the cytoskeleton is going to move the vesicle to the edge of the membrane, and when the vesicle comes close to the membrane, the two phospholipid bilayers are going to fuse together and eventually release those molecules out into the environment. So this involves a lot of energy. The cell has to move the vesicle to the edge of the membrane, and then the phospholipid bilayers fuse together to release the particles outside. The other example of bulk transport is endocytosis. Endocytosis. And endocytosis is bringing particles into the cell. And there are three basic types of endocytosis. There's phagocytosis, pinocytosis, and receptor-mediated endocytosis. Phago means to eat, so this is where the cell brings in a large food particle, or it could even be another cell. And for this to occur, the cell membrane invaginates, and then it pinches off. So these two ends, or these two sides, come close together. It pinches off, forming a vesicle with that food particle inside. pinocytosis. Pino means to drink, and this is where you have very small particles or liquids that the cell takes inside. So now this is a solution rather than a solid particle, but the same idea where the cell membrane pinches together, pinches off here, forming a vesicle that then moves inside the cell. The last type is receptor-mediated endocytosis, and this is triggered when a particle, it could be a small particle, could be a large particle, but it binds to a receptor, and then that triggers the cell to perform endocytosis. So bubbling, bringing in the membrane to form a vesicle, pinching it off here, and now you have a vesicle with that molecule bound to the receptors. So bulk transport can involve releasing molecules or taking them in, but it involves moving the cell membrane. So that involves ATP and the cytoskeleton forming vesicles to either bring molecules in or fusing vesicles with the membrane to release molecules out. This is the end of the lecture on Chapter 5 for membrane structure. In this lecture, I went over the different components of the cell membrane, the different functions of the cell membrane, and different types of transport across the cell membrane.